Welcome everybody to Discovery Days in Health Sciences here with the Queen's University Faculty of Health Sciences. We have the keynote lecturer coming up. Dr. Jane Philpot is the new dean there at Queen's University. She was unable to be here today. She really wanted to do an intro, but she did take the time to create a video. So if you bear with me, I'm just going to play now her intro video.
Hi, everyone. Good morning. On behalf of Queen's University, it is my delight to welcome you to the Canadian Medical Hall of Fame Discovery Day. My name is Jane Philpot, and I'm the Dean of the Faculty of Health Sciences here at Queen's. You're here today because someone at your school thinks that you would make a wonderful healthcare professional. That's a great compliment. So let's start our day by recalling how special it is that you were chosen to be here today. Becoming a health professional is a wonderful calling. We need the brightest, most dedicated, and compassionate young people to choose to study the health sciences so that you can take care of the health of the people in our country. And today we want you to get excited about the possibility of taking that on, about the idea of a career as a health professional. Here at Queen's University, we have a special arrangement where we have a Faculty of Health Sciences that has a School of Medicine, a School of Nursing, and a School of Rehabilitation Therapy. This gives our students a fantastic advantage in order to learn and study and care for people in an interprofessional way. That's a key to becoming an exceptional healthcare provider. If you're looking for a career that has versatility, health sciences is for you. You might become a surgeon, you might become a health researcher, or someone that cares for people with mental illness. There is a realm of possibility. The format might look a little bit differently in the future in terms of how we do things in health sciences, but today is your chance to explore the possibilities, and I encourage you to keep an open mind as you move through the day and see what sparks your interest. So now it is my pleasure to introduce you to Dr. Lynne Postovit. Dr. Postovit is the department head for biomedical and molecular sciences. In addition to being one of the biggest departments in our faculty, in addition to leading one of the biggest departments in our faculty, Dr. Postovit is also a world-class researcher. Her research focuses on cancer, specifically ovarian cancer and breast cancer. She is not a medical doctor, and I mention this because when you're contemplating your future in the health sciences, you don't need to be a medical doctor. You can be a basic scientist, a PhD, like Dr. Postovit. She does all the background work that's necessary for future breakthroughs in scientific discovery. Some people undervalue the power of health research, but there's an enormous amount of work that goes into research labs, and the work in those labs is absolutely essential for the major medical discoveries in life. So when you hear about a new cancer drug or a new antibiotic or a new vaccine that saves lives, you can bet that the discovery behind that was led by a team with someone like Dr. Postovit in charge. Someone who has spent years dedicated to working in a lab, looking through microscopes, working with data to get a better understanding of our body, our cells, and our molecules. Eventually, that kind of work can lead to the discovery of life-saving medications and other forms of treatment. And so with that, I pass you over to your keynote speaker, Dr. Lynne Postovit. Thank you. I hope you enjoy your day.
It's a wonderful intro, and I want to thank Jane for that, and I'm going to try to share my screen. It's certainly a pleasure for me to be here with you, and I'm going to attempt to do this. I'm a little technologically challenged at times with some of this, but I hope that you all see this. And let me know if you can't, I guess, because I can't see you through this. So today I wanted to take you all on a bit of a journey from the place where I think you're sitting at now as students in our high schools, all the way to where I'm sitting now as the head of the Department of Biomedical and Molecular Sciences here at Queen's, and a professor as well as a research scientist. And so I titled my talk thinking back to how it was back in the 90s when I was around your age too. I did not know what I wanted to be, but I just wanted my life to have meaning. So you've heard a little bit about me, but I think I thought I would start by just adding to that so you can get a sense of who I am and where I've been. I just moved back, and you'll hear about why back, to Queen's University this last year, really, from the University of Alberta, where I was a co-director of a cancer research institute and also endowed research chairs in the areas of ovarian cancer. I am a researcher in the area of cancer research, and as Dr. Philpot said, we have a particular focus on cancers affecting women, and so these are breast cancers and ovarian cancers as well as endometrial cancers. And I made the move back to Queen's to really make a difference, and I saw a tremendous opportunity to do that within the Faculty of Health Sciences. And as I'm going through this talk, I hope you can see that it's really an environment that kind of intertwines education and treatment and healthcare delivery, as well as research. And I think that that's an important movement forward as we're moving into a time of great change, and I think tremendous potential. And this is because there are tons of enabling features. So we work in very collaborative teams within the faculty as well as in the department. We have great abilities to run clinical trials and to translate our research. The infrastructure is great, and importantly, we have a community. So beyond that and the nuts and bolts, that the aspect that really drives the research in our laboratory, and I say our because it's really a team made up of amazing people from around the world, are cancer patients. And so in our case, it's our sisters, our mothers, our friends, teachers, colleagues, women in our community that have suffered cancer and often lose their lives all too quickly. And so this really drives us. We work a lot with patient groups to try to understand their needs, and also to remind us of the important work that we do, and to keep us motivated when times and questions are hard. And so our goals from a research perspective are really to improve statistics associated with cancer. And we're doing our part by trying to define factors that can treat and predict advanced cancers. So in the laboratory, we're really looking at those cancers that are resistant to treatment and that are spreading. And I'll get to that. So we're going to begin the voyage almost 30 years ago. And I don't know if you guys know Where's Waldo. So it was popular book certainly when I was a kid, but this is kind of what we're going to play through this talk. So where's Waldo, except where's Lynne? And we'll see if you can see me in this one. But this is a picture taken probably in the early 90s, early to mid 90s. And you can probably tell by the outfits and the archaic televisions and that type of thing. And I put it up here just to show you that I was just like you. So I'm a young girl here with her friends. I grew up in Whitby, Ontario. My mom stayed home. Dad worked at General Motors. I would say it was a pretty standard life. And at this point in time, I had really no clue what it was that I wanted to do. And of course, in high school, in grade 10 or 11, they always have you write an essay about what you're going to be, I guess, when you grow up. And this is actually an excerpt from that essay that I wrote. And I said, as an adult, I will be a Pulitzer Prize winning author. I will be a scientist who finds a cure for cancer, and I will be the first elected female Prime Minister of Canada. And I actually had to write in elected because our first female Prime Minister is, of course, the Honorable Kim Campbell, but she wasn't elected, so I added that in. And it's kind of hilarious because it's probably the essay of a massive overachiever, but it really was because I had no idea what I wanted to do, but I did want to do it extremely well, and there to be meaning to the work that I would do. And so after high school, I actually went to Queen's University, and it's where I've actually come back home to. And I again didn't know what I wanted to do. I was really into poetry, hence the writing, and politics, but also science. And so I thought, well, I'm gonna start in science, and had a lot of interest in physics and mathematics, and then also the life sciences. And so I kind of meandered through and tried to find what was best for me and what I wanted to learn. And what I learned in this time is that really universities are a very special place. And so above all, they're a place where we disseminate all the knowledge of the world, and you can have access to all of it. It brings, of course, societal enlightenment and training. But the most important thing to me was really being able to access brilliant folks from around the world, as well as students, and to learn deeply about a number of different things. And I ended up being in the life sciences program, which is still going on here at Queen's. I began to appreciate that it wasn't just sucking in this knowledge and integrating it that excited me, but I wanted to be a part of generating knowledge, and really universities are a place where that occurs. So we take all the information in through the dissemination and through the learning, and then we toe the very edge of what is known, and then we actually, as researchers, take that next step into the unknown to generate knowledge. And one of the best ways to do this, if you are enthusiastic about it, is by pursuing a PhD, and that's exactly what I did with Dr. Charles Graham in what was then the Department of Anatomy and Cell Biology. And my project, and I'm going to say this last 5 years, so it's going to take a long time, related to comparing to cancer, and something that was common between the two of them, which is the levels of oxygen levels that are available in these scenarios. And so what we knew is that with probes we could go in, and we knew that low oxygen levels characterize the time of implantation and early blastocyst formation, so the very beginnings of the embryo and the formation of the placenta, and that this actually occurred for the first 8 to 10 weeks of pregnancy. And so something about that we thought was probably pretty important for setting up the pregnancy and making that placenta. We also knew that hypoxia characterized the microenvironment of solid tumors. We had a lot of low oxygen in there, and that when tumors were hypoxic or had low oxygen, they were more resistant to therapy, and they also seemed to spread more. And so we looked at these two things and said, well, what might be common between them? And one of the common aspects is the ability of the cells to invade into a tissue. And so here I show you an amazing photograph of a macaque monkey blastocyst into the uterus. And kind of that tissue that looks like clouds, that's actually going to be setting up the placenta with a cell type called a trophoblast. And then the embryo is kind of the disc-shaped structure at the top. And the main thing to look at here is that, well, this is a situation that's very low in oxygen, and these trophoblast cells have to invade a tremendous amount in order to set up the complex nature of the placenta. And in cancer, we actually see something similar occurring. And so here's sort of a cartoon of what we would envision as a breast cancer. So in a breast cancer, we would have tumorigenic cells that actually fill the ducts within the breast, so almost like a sausage, if you will. They're surrounded by what we call a basement membrane, kind of like a casing, and the blood vessels are on the outside. So if you can envision this, as the cells begin to fill this tube, they have less and less access to oxygen as they're coming through. And at one point, they begin to invade out of that basement membrane and into the tissue. And that's when we know cancer becomes more difficult to treat, that invasive process. And so we put these two things, low oxygen is playing a role in how trophoblast cells of the placenta, as well as breast cancer cells in this case, are actually invading. So we did a lot of assays in the laboratory with cells under microscopes, counting them, etc. And what I really want to point out here is that this was in fact the case. So those red bars show that low oxygen levels increase the ability of trophoblast cells and cancer cells to invade. And we figured out that this was by upregulating a protein called urokinase receptor. And so you have one finding that's super important, and then another one that adds to it and increases your understanding of what's going on. So we want to see if this might actually be important for cancer. And to study cancer, we usually move from cells into different types of animal models. And in this particular model, we were looking to see whether cancer cells could grow in the lungs of a mouse. So these are breast cancer cells, which when they metastasize often go to the lung. So we put them in there, and then we quantify what's happening. And what we did before putting the cells in is or normal conditions, or even some factors that we thought might be mediating the process. And what we found, and as you can see here in this time with the blue bar, is that if we expose cells to low oxygen levels, they actually metastasize more. And then you can see here something called GTN and D-to-nono-weight, and these are some of the mechanisms we were getting at. So GTN is glycerol trinitrate. It's actually what's used to treat angina, and you can see that actually prevented the upregulation in metastasis. And we were able to figure this out with lots of molecular biology and understanding different pathways, and showing that nitric oxide in some instances could actually mimic oxygen, and so prevent some of the effects that we saw when we took oxygen away. So after 5 years, and I love that I can summarize all of this in one slide, we actually generated knowledge. So we determined and validated that low oxygen levels do promote cellular invasion, and that has been pursued by laboratories around, including my own, in all the way up to now. We also determined that this occurred through oxide signaling, which allowed us to determine that if we resupplemented nitric oxide with things that patients were already using, like GTN, we could actually prevent certain aspects of cancer progression. And these findings actually led to patents, a startup company, and clinical trials where we were testing the efficacy of glycerol trinitrate in prostate cancer patients with some improvement. So it was really exciting. And I want to highlight it to get you all excited to show that a simple set of experiments done by a student in a university can actually make a real difference. So when one finishes their PhD, this is probably the most important one. So in addition to being in the laboratory and learning lots and absorbing information and generating knowledge, science and actually all of the medical professions are incredibly collaborative. And this is a picture here taken probably 1,000 or so. And I highlight it because it actually has 3 generations of scientists. So can you see me? So where's Waldo, or where's Lynne? I'm there. There's another student, Nicola, that's there. And then there's Dr. Adams and Dr. Graham, supervisors, and Dr. Piyush Lala, who was the supervisor of Dr. Graham. So 3 generations of scientists. And the other thing is that science is super global and actually very fun. So this picture was taken in New Orleans at an amazing restaurant called Commander's Palace. And I will say that in grad school, it was the first time I even went on an airplane and began to see the world to share my science with others around the world. Okay, so the next step after the PhD is the postdoctoral fellowship. And this is really almost like an apprenticeship. So PhD, you've generated knowledge, you're mentored, you're prepared, and then in the next phase, you're moving towards being able to run your own laboratory and to help mentor others toward that same goal. So I did my postdoc at Northwestern University and Children's Memorial Hospital in Chicago, Illinois, and it was quite a move from Little Kingston to big downtown Chicago. And I worked with Mary Hendricks there, and we were asking, again, the question of how do cancer cells resist therapy and metastasize? And at that time, we started to think about cancer as being highly adaptable. Just a little cartoon that tries to illustrate that. So it says that a tumor is actually very heterogeneous, but within them, indicated by red cells here, there are cells that acquire the ability to adapt so they can better resist therapy and they can spread more. And we thought, well, these are the actual cancer cells that we want to target. And since then, we've changed the name of them. We call them cancer stem cells, etc. But really, we're very interested in understanding how they are the way that they are. And we explain this with the concept of cellular plasticity. And so plasticity, in the case of an organism, this is an evolutionary concept, is the ability of an organism or cell to change phenotype in response to an environment or a microenvironment. And we think of cancer in the same way, where plasticity, or that ability to change, would be a huge selective advantage, allowing the cells to move around the body and metastasize, and then also to resist therapy. And I have in the background here a picture of an African butterfly, and this butterfly exhibits tremendous plasticity. So when it moves from its home in Africa up north into Europe, it actually can change a bunch of genes that it expresses and then acclimate to the colder climate in Europe. So this is a concept that's evolutionary that we're going to now apply into cancer. So what does this look like in cancer? Well, it turns out that as cancers get more and more aggressive, they look less and less like the tissues from which they come. They actually look more and more embryonic. And then they begin to behave differently. They're able to survive more, what we call self-renew, which is just keep on repopulating themselves, and importantly, to adapt. they can even start to look like other tissues. And so sometimes cancers can actually look like the endothelial cells or the lining of the blood vessels. They can sometimes even look like structures such as bone. So they change a lot. And so this is basically the movement between different cellular fates. So big question, and this has actually been something that now I've been asking for 20 years. How do cancer cells acquire and sustain plasticity? And to understand this, we actually use a landscape, the Waddington's epigenomic landscape, a really, really long time ago. And then this was redrawn and redescribed by Dr. Yamanaka, who won the Nobel Prize a few years ago for induced pluripotent stem cells. And it looks pretty complicated, but essentially what it says is that if we take an embryo and the cells of an embryo, so those embryonic stem cells, they have the ability to become anything in the body. However, as the cells move through development and they become the various cell types of the body, it's almost like going down a hill and then getting caught into divots. So if you look at the bottom, we would have, say, skin cells and gut cells and brain neurons, etc. And the idea is it's really hard to go back up the hill and become pluripotent again, and it's just as hard to move from one cell type to another. And of course, Dr. Yamanaka showed that maybe it isn't that hard. We figured out what the factors were to push it back up the hill to that totipotent state. And we think there's something about cancer that also allows this type of flexibility or promotes it. And so the lightning bolts are actually a key component of cancer, which are mutations. And so the DNA gets altered, mutations occur, and we think perhaps something about these mutations makes it a little easier to go back up the hill or a little easier to look like other cell types. And so this is just another way of showing the exact same thing, where we've placed cancer on a bit of a continuum. So we have embryonic stem cells to the left, we have differentiated tissue in the middle, and then we have cancer to the right. And the idea is that cancer actually adopts a lot of features of these stem cells, with the key difference being that they're no longer regulated. So stem cells, of course, can always turn into the right tissue. Cancer, we think, maybe doesn't listen to those cues so well. And I'm only showing you this because it's telling you that maybe the key to understanding cancer and why it's so plastic is actually by understanding development first. And this is exactly what we did. So we thought, well, if cancers are behaving like stem cells, maybe they're using stem cell-like factors. So if we were to take cancer cells and put them into an embryonic environment and see what they did, we may be able to move back and figure out how they themselves are becoming like stem cells. And so I'm showing you this because it's probably the favorite experiment that I've done so far in almost, I guess, 25 years that I've been doing this. And this experiment actually used a model called a zebrafish. So they go around in fish tanks, but they're also an amazing developmental model. And that's because we understand how the fish develops. It develops within 24 hours, and we know how to change that and what the factors that might change that would be. And what I did is I labeled cancer cells green, and I injected them at the top of a zebrafish blastocyst, or what we call the margins, so near where the yolk is, and just look to see what happened to the fish in development. And this is what happened. And so we took cancer cells C81-61s, that's on the left-hand side, that don't form metastases and don't kill the patient. So these, I guess, would be good cancer cells. You take them out, the patient's fine. And then a version of those cancer cells that metastasize everywhere and are extremely difficult to kill. And what you can see is that the aggressive cancer cells made a bump on the zebrafish, and the non-aggressive ones really didn't do anything. So we did some imaging. This is something called confocal. All you need to know is that the green cells are cancer cells. The red outlines are the outline of the zebrafish. And you can see that the cancer cells didn't just make a tumor on the fish head. It actually organized a structure. And if we injected the tumor cells closer to that egg, like I told you, and then we let the zebrafish grow a little longer, what we ended up seeing was a two-headed fish with the cancer cells at the bottom. And these fish would go around in a circle, And the exciting thing about this was that it was described years and years before by Schmiemann and Mangold, so classic embryological experiment, where they took what we call the dorsal, their backside of one frog embryo, and put it into the front side of another frog embryo, and guess what they got? A two-headed embryo, just like this. So it was a duplicated axis is what it was called. And the really great thing about it is there aren't a lot of things that can actually do this. So this crazy experiment allowed us to discover that cancer cells make a protein called Nodal. And this was the first description of Nodal in cancer. Nodal is an embryonic morphogen, so that means that it actually helps to instruct how embryonic tissues are moving and what they're doing. It initiates what we call the embryonic axis, so that's essentially your backbone from top to bottom, as well as what's left and right in an embryo. It is expressed in these cancer cells, and we now know that its downstream signaling changes them completely so that they're able to be much, much more plastic. And so here's the next picture of Where's Waldo, and it's just showing the importance of mentorship here. So to the left is Dr. Mary Hendricks, amazing lady, my postdoctoral supervisor, who's now the president of a university down in the United States. And then on the other side is Charles Graham, my PhD supervisor, and the two of them really helped me to be the person, I guess, that I am as a researcher. Okay, so big discovery done. It was time to start my own lab and taking the knowledge that I had generated. And my first laboratory was at the University of Western Ontario in beautiful London. And here's a picture of that here. Add to that a little bit. So we've got Mary, Charles. Add these two young ladies because they were actually my first students. So a huge part of this job is actually mentoring and training the next generation to generate knowledge themselves. And Daniela, who's the first student, is now a professor at McGill, and Megan is a lawyer. So it's been amazing to see their careers evolve as well. So we made this big discovery in Chicago, and we went around the next 5 to 8 years just asking the question, what happens when we target Nodal in tumors? And the answer to that is probably best illustrated by this image from one of our papers, which is that they grow less and they're easier to kill. So this is just an image of green, in this case, breast cancer cells with red blood vessels moving through them. If we take away Nodal, these tumors really don't form. The blood vessels don't move in. They don't evade the immune system as well. They don't spread as well. They don't resist therapy as well. And we've shown this in melanoma now, breast cancer, and ovarian cancer in the lab. So what we've learned is that Nodal can be detected in a variety of cancers, and in all of them it seems to do really bad things. So it's associated with a poor prognosis and resistance to therapy, etc. We have one exception that a graduate student, a PhD student in the lab, Paula, is working on now, and that's thyroid cancer. So that's kind of interesting too. We find that Nodal, because it's secreted, not only regulates the cancer cells, but all the cells within the tumor, and changes how they're able to interact with the tumor, making the tumor progress even more. And importantly, we're working with chemists and companies to develop Nodal inhibitory peptides and proteins that we maybe be able to use in patients to help prevent plasticity. So pretty exciting, an evolution going there. And so while I was at Western, I did another thing. So I became a mom. And so this is just a picture of my son at about 10 months old. And he isn't a duck, he is a human, but that was him there. So the next chapter, I was recruited to the University of Alberta in Edmonton. And that was quite a move for me, for coming from the East. I'd never lived further west than Chicago. So it was an amazing move, and I was recruited there to start to begin to focus more on ovarian cancers. And I'll give a big thumbs up to University of Alberta, amazing facilities, great university, and tons of medical research occurring there. And so in this next chapter, we are kind of moving forward and asking, how is it that these stem cell factors get turned on? And I'm not going to get into this a lot because it might make your head spin. It's for later, but essentially what we're finding is that stress itself, so going all the way back to my PhD and things like hypoxia, actually cause the proteins like Nodal to be produced in a higher rate. And once they get produced at a higher rate, they start to reprogram the cells to become more plastic and more adaptive. And this is really interesting because it's taken us now beyond one factor like Nodal to be looking at a whole cohort of factors. So looking at that upstream regulator, what we call integrated stress response, may be the key to actually eradicating a whole program of plasticity. And it's something that my lab is really focused in looking at. So what we learned in this time is that stresses like hypoxia promote cancer cell plasticity. We know that plasticity is enabled by alterations in how cells make proteins, and what we call the translational machinery. We can prevent adaptation by targeting pathways that regulate protein production, which is really cool because now we can target a number of different pathways. And we've also just found a lot of crazy things, and a lot of that's been actually enabled by technology. So in this last year, for example, we've seen a virus be identified and then a vaccine made, and that's because we could sequence it, produce the protein, mRNA vaccines, which were based on years and years of amazing research, and we're harnessing that in our lab too, from imaging to molecular biology to sequencing. So now we know the DNA looks different when cells are stressed. When cells are stressed, they can evade the immune system, and we can start to look at how that's occurring. We think that plasticity may be preventable, and that it is probably induced very early on in cancer. So how do we actually prevent that from happening? And so this is the evolution. So the duck became a mad scientist, and here he is in Edmonton. And I think another important point is that as researchers and health professionals, it's a part of our life. And so we bring our life often into our work. So he'd probably kill me now for showing that. So chapter 3, now that I've moved back to Queen's, what are we doing now? So in addition to now mentoring my laboratory, about 15 people, brilliant folks from all around the world. I'm helping to also lead a department of amazing scholars doing all sorts of types of research. Our own questions, though, are all around what I've just described. So how do mutations enable plasticity? How does DNA change during stress? How do the changes in DNA actually drive plasticity? And can we change the DNA to actually affect how different types of proteins are made? And most importantly, how can we prevent plasticity from occurring in the first place? So all of this takes a massive effort, I'm not gonna lie, but it's made more pleasurable and more powerful by collaborators now around the world. We can Zoom at any time. One benefit of Zoom, or these online modalities, and importantly, really driven by brilliant students such as yourselves that come into the laboratories and really want to generate knowledge. So to conclude, I hope you appreciate that basic science can make a huge difference and is actually integral to advancements in medicine. So by understanding level, we've actually been able to discover new therapies that are used to treat people. Meaning is derived from the process, not the endpoint, and I hope I articulated that. And you should never lose sight of the importance of the journey. So sometimes we're thinking in our minds of who we're going to be and what we're going to be, and really the most important parts are the bits along the way that bring us to that point. Knowledge beyond everything is power. If I have one piece of advice, take in every little bit that you can, and take it in for the sake of taking it in. You never know when you're going to use it, and if you ever want to generate new knowledge, you must first understand what is already known. Medical advancements evolved from a discovery made by a student or a colleague affiliated with a university. And universities they just don't educate. I mean, that's certainly a massive function of them. They importantly generate the knowledge that advances our society. And the world is small and it's getting smaller. So teamwork is just so very, very important. We all have to respect each other and to embrace our ideas. And truly, we can do so much more, so much faster if we just work together. And with that, I'll exit and I guess take some questions.
I was just talking briefly about how all of this incredible contribution to health science is going on in the background while we sit here and are mostly unaware of it. People like Lynne, in this small sliver of a discussion, we're learning there's thousands of people like Lynne trying to solve these gigantic health problems and making progress in solving these gigantic health problems. So very much appreciate your time. It's very clear how busy you are, and the fact that you're able to give us some of that time today is truly, truly appreciated, Lynne. We really are pleased to have you join us. Thank you so much. Appreciate you being with us. Hope you I certainly am inspired by your talk, Lynne. I think this is a great way to start an important day of discovery. Thank you so much once again. And students, we hope that you enjoy your day of discovery. Have a great one.